So this slide starts us off with a look at mus musculus as a model system, primarily for modeling how things might work in humans. So I highlighted down here, it's a model for testing out drugs, for testing all kinds of things, first in mice to get some idea of how it might work in humans. Because the mouse has a lot of advantages, they are very closely related genetically to humans. They are mammals. Of course, chimpanzees are much more closely related, but chimpanzees don't make a good model organism. Mice are small. They reproduce quickly, really just 12 weeks from one generation to the next. They're only pregnant for 21 days. And then when those babies are born, there's just another 9 weeks until they're fertile. And so it's very quick. You can get a lot of offspring in a very short period of time. They can be inbred, all kinds of things. In fact, there's a lot of demand for people who can work in a mouse room on a university campus to handle all the mice that are being used in different testing and experiments. So anyway, genetically down here, I show you how many of the genes are the same. Doesn't mean that every nucleotide is the same, but they have 99% in common with humans at the gene level. And their diploid number is 40. Of course, our diploid number is 46. So similar enough, although once you test something in mice, there's still a chance when you go to human trials it won't react the same way, but it is a first level of testing that we do with anything. And so as far as biotechnology goes, transgenic mice, they started to generate transgenic mice very early on in the 1970s, I think, 1980s. But the introduction of this special technique where you could actually hold an egg still and then a very, very skinny needle that could actually inject DNA directly into the nucleus of a fertilized egg. This technology is still used, and it's just really amazing. And you can then implant those genetically modified eggs into typically a surrogate mother. And then the babies are born out. When they're first born out, you can't tell which ones of those embryos may have actually integrated that gene into a chromosome. So you do a little PCR test, and maybe you find out that pup A and pup C and pup D show evidence that trans gene is now in their DNA. And so you can grow them up and then mate them and so on and so forth. And so all kinds of schemes for creating mice with certain genotypes, with certain transgenes. And really, transgenic mice, it's just a gene delivery, right? It's just like we did transformation of bacteria. We delivered DNA into a bacteria, and the bacteria then takes on that genotype and then maybe a new phenotype. So you can use transgenic mice for all kinds of things. But this was kind of a first level of manipulating mice by adding a gene. What has happened since then is they've been able to modify the procedures to where we can remove genes. So there's a procedure like that, and that we call those knockout mice. And then now with CRISPR, you can target a mutation to a particular gene. But these are all based on the same kinds of technologies, just improvements and tweaks and little changes that are added, little features, I guess you could call them. So when we started messing with mouse transgenics, certainly people were thinking in terms of humans. Now, when you inject a gene into a mouse embryo, you're changing the genetics of not only that mouse, but all of their descendants. And so that's called germline transgenic, meaning you're permanently changing the lineage of that mouse and their descendants. In humans, that is not something that we do. It's not, in the US, that is not something that is allowed. But we are doing gene therapy where we deliver genes to adults. And we only deliver genes to somatic cells, not to the germ cells, not to the reproductive cells. So currently, gene therapy treats somatic cells only, so somatic gene therapy. If we were doing germline gene therapy, you could do DNA injections into embryos, but it would be human embryos. And that right now, there's no, that's not allowed in the US. Can't say what's allowed in other countries, right? So this is just a gene delivery. The first gene therapy was done with virus vectors, meaning a viral chromosome and then a human gene of interest were attached together. And then that construct is put into a virus particle, and then that is used to infect cells. And so did all kinds of different versions of this, starting in, I'd say, the 1990s. And the first trials were very kind of controversial, as you can imagine. The two main types of vectors that were used at first anyway, one was an adenovirus or a herpes simplex virus, where they just removed what they considered to be the kind of harmful viral genes and then integrated the human gene of interest. The thing about those viruses is when they inject their DNA into your cell, the DNA stays as what we call an episome, not integrated onto the chromosome. So once that cell dies or divides, then that DNA or that genetic material is not kept. It doesn't last. So you get kind of a transient gene expression that sort of fades. And so you would have to re-dose the patient kind of on a regular basis. Then you have retroviruses. That's a virus that integrates onto chromosomes. So that's more potential for a stable long-term expression as the cell divides. But a virus only has access to chromosomes during mitosis. In prophase, that's when the nuclear envelope disappears, and that's when the virus can integrate onto a chromosome. And so not all cells that are targets for gene therapy are actively dividing tissues. So retroviruses aren't always possible, depending on what the application is. But here's just very quickly some of the early ups and downs of gene therapy. I mentioned before, there was a patient that was cured of SCID. SCID is severe combined immunodeficiency. It is the failure of your immune system to mature after you're born. And so you live and this was a child, but you live all the way until you die of some infection. You live with really no immune system. And so it's very dangerous. In fact, this was what we call the boy in the bubble disease. David Vetter was the first, I guess, SCID patient that they actually built him like this, what they hoped was sterile living environment inside a plastic bubble. And but he did eventually die of some simple infection. So, but Ashanti Da Silva was the first one that was cured by gene therapy because they delivered the gene that she had two mutant copies of. They were able to deliver a good copy of that gene into enough of her white blood cells or bone marrow cells that she could then start maturing her immune system, and it developed. So that was a success. Then there were a lot of trials that were kind of nothing great, but nobody was harmed. In 1999, though, there was a famous case where a young, I think he was 18 or 19-year-old patient died when he participated in a trial of gene therapy for a disease he had called OTC, ornithine transcarbamylase deficiency. And he was controlling his disease with diet, because it's a metabolic pathway of amino acid metabolism. And so he was controlling his OTC deficiency with dietary restrictions, but he wanted to participate for people who had worse, cases of the disease. And so he participated out of the goodness of his heart, but after he got an injection of the gene therapy, he died. He went into multiple organ failure. And it turned out that they didn't remove enough of the viral genes out of the adenovirus, and he had a bad reaction to that. So that was really hard. They shut down a lot of the trials for different kinds of gene therapy at that time. And then things started to kick back up again. Around 2000 and 2001, they continued on with some of these SCID experiments, the severe combined immunodeficiency, and they were able to cure 10 out of 12 patients in one of the trials. But in 2002, it turned out that 2 of the 10 cured patients eventually developed leukemia, and they knew it was the result of the gene therapy. They were able to go in and see that the transgene had integrated next to an oncogene which causes leukemia, and that had stimulated overexpression of that leukemia gene. And so, we have this, what I call ups and downs. I mean, we definitely have had some success in these early years, but then, one of the rules of medicine is do no harm. And so we definitely did harm. And this statement here, I need to change this. Up till now, we actually have gene therapies that have now been approved. So you can cross that out. There's been several gene therapies approved. You're going to do one you may have already finished in one of your homeworks for hemophilia. There's definitely some in the works or maybe recently approved for sickle cell anemia, some for certain blindness, inherited blindness type disorders. But anyway, you can cross that out. We do have approved gene therapies now. I forgot to remove that. I need to. All right. So just to show you how the SCID was cured, what they do is, and this is kind of a general scheme, they take a good copy of the gene that's missing, that's mutant in the people who have the disorder, and then they recombine it with a viral vector, and they create this virus that contains the gene of interest here in blue, and then they infect cells from the patient. So they take the cells out of the patient. It's pretty easy to get bone marrow from a patient, and that's where your stem cells are for your blood, for your white blood cells. And they can just infect those cells in the Petri dish and wait for them to divide. And when the cells divide, the gene goes into the nucleus and becomes integrated onto one of the chromosomes. At this time, they cannot target where the gene integrates. And so then they reintroduce those cells back into that same patient. The genius of this is that you won't get rejection because the cells that are being genetically modified or genetically corrected are from the patient. When you introduce them back in, they will be accepted. There won't be any immune response. The thing they did wrong was they needed to check the cells prior to reimplanting them because some of these cells eventually became cancerous. And so they needed to come up with some way to screen, and that means look through the cells and test them or look at whether or not the gene integrated next to an oncogene. So after this happened, of course, they were aware of this. I'm going to argue they were kind of aware of it. I don't know where the failure actually happened because this wasn't such a surprise when it happened, but I wasn't involved in that trial at all. So, but anyway, so they, of course, they learned from that mistake, terrible mistake. And the last slide is the kind of the newest of the technologies, this CRISPR-Cas9. Now, CRISPR-Cas9 is sort of, it's maybe the newest technology you know of, but it's sort of now there's next-gen CRISPR. There's other kinds of CRISPR that have already come out. But I'm just going to show you kind of the vanilla plain CRISPR, and different iterations have already been developed. So what CRISPR is, it's a naturally occurring system in bacteria. Bacteria have a couple systems for protecting themselves against phage infection. We talked about restriction enzymes, which cut up a phage chromosome as it comes into a cell to prevent infection. But CRISPR, once a chromosome comes in and the restriction enzyme cuts up the DNA, what the cell will do is actually save a piece of that phage DNA and insert it into its own chromosome. And so it turns out that bacterial cells keep a little piece of every phage that has ever infected them. And then if that same phage infects them again, what they will do is they will express that gene that they kept from the last time that phage infected them, and they will combine it with an enzyme called Cas9, which is an enzyme. And so the RNA from the previous phage becomes a guide, and it sticks to the Cas9, and the two of them together go through and attack any DNA that is complementary to the guide, which would be the newly infecting phage. And so it cuts the DNA that is coming in from the phage. So there's basically several ways that the phage DNA is attacked. One is just from restriction enzymes, and the other is from these saved pieces of previous phage that are then expressed and combined with a Cas9 enzyme, and that becomes a seek and destroy unit inside the cell, which destroys the phage. But what humans have figured out is you can create a guide any way you want, and if you add a second RNA, you can cause the Cas9 enzyme to not only cut DNA, but introduce changes or insert DNA. So there's tricks that we have figured out that Cas9 will do if certain synthesized RNAs are provided. And so by combining these synthesized RNAs with the Cas9, you can target, you can create mutation, but you can also correct existing mutations, and you can actually copy in whole sections of DNA into a target site. So this is a very powerful technology. But like I said, this is being used on somatic cells, not on germline cells. So we're not doing human gene editing in eggs or sperm or zygotes, although it's totally possible to do. It's theoretically possible.